Okay, I'll just introduce our next speaker. So again, I apologize that we're a little bit out of alignment with our scheduled speaker list. But our next speaker is Sina Mansour from the University of Melbourne and the National University of Singapore. So please join me in welcoming.
Hello everyone, I'm delighted to be here today presenting our work on spectral normative modeling and its application for high-resolution brain abnormality inference. So before everything, I have nothing to disclose, and with that out of the way, let's talk a little bit about spectral normative modeling, which is basically the method sitting behind the results that I'll be later sharing in the second half of my presentation. So it's essentially a method that kind of connects two different realms in neuroimaging, the realm of normative modeling to the realm of brain eigenmodes. So let me first talk a little bit about each of those. Normative modeling is essentially the idea saying we can characterize population-level phenotype distributions and detect individual deviations from what's considered to be normal. It's kind of similar to how pediatric growth charts work, but it's with brain imaging data. So we get scans of brain images of individuals, and we have a phenotype of interest, let's say it's cortical thickness, and for a new scan that we collect, we can put it against the population scans that we have collected before and kind of guess or estimate the centile that that individual is, saying if the individual brain is normal or abnormal according to their demographic profile. And this method is kind of interesting because of the unique features that it comes with. It essentially enables exploration of individual level differences, which is something that is missing from the traditional group level versus, so healthy control versus patient kind of studies. Essentially, we could dive into investigating the way in which a pathology affects the brain. It might be that all individuals with a pathology show the same pattern of abnormality in their brains, or there might be subtypes, so different individuals have different patterns in which their brains are look different from normal, and there might even be more complicated cases like a gradual decline or gradual deviation from what's normal, or comorbidities introducing complex heterogeneity in the final patterns of abnormality. And traditionally, when we want to do normative modeling, we start with a fixed question. So this is what I call a fixed phenotype of interest, and I call it a spatial query. Basically, we want to, so the spatial query, to give you an example, can be like the cortical thickness of a specific part of the brain. And we essentially want to find a model that explains the distribution of that variable across our population against a set of base covariates, things like age and sex. And the way we do it, we basically collect a large sample of imaging data from the population, hopefully covering a long age range, maybe the lifespan. And then we have our fixed query predetermined. We let the model run, and eventually it gives us the parameters. Later on, when we have a new scan, we can just compare the new scan based on the brain charts that we have computed, and that is going to tell us that whether that individual is having an abnormal brain or not. And looking into this, we kind of noticed a gap, and we were thinking, would it be possible to separate that step where we define a query from the step that we kind of fit the normative model? And this is basically the idea of the method. We use graph spectral decomposition ideas in order to compress the normative information that we collect from the brain imaging data so that the query can be flexible and determined at the latest stage, not at the stage that the model is being trained. And for this purpose, we're using brain eigenmodes that were extracted from a group-level vertex resolution structural connectome of the human brain, and we collected many brain eigenmodes. As a parameter, we tried to see how many eigenmodes are needed to be able to capture all the information about the normative trajectory of the brain. Essentially, the way to look at this is we have a filter or a way to summarize information, high-dimensional information on the cortex of the brain into a latent space. And later on, the way the normative model, the spectral normative model would work is we don't need the query in the training step. We just learn the loadings onto different eigenmodes and how they change across the population and across the lifespan. And later on, when someone wants to use the model, they can provide any spatial query that they are interested in. And we essentially, first of all, compared the performance of our approach, and we saw that if we include at least 1,000 modes in the model achieves accuracy comparable to the traditional normative models, albeit at a relatively faster and more efficient computation time. And we tested it across a range of different possible spatial queries, so things that could be global, let's say the thickness of the left hemisphere, and things that could be regional, like the thickness of a special region defined by an atlas, or even a high-resolution vertex-based query, which is one of the utilities of spectral normative modeling. We essentially can go to the high resolution and efficiently estimate the normative ranges at vertex resolution. And what that means is that we get a brain chart for the human lifespan that is specific to the vertex resolution of the brain, or essentially that is at the resolution of the imaging data collected. And what this is useful for, one of the examples, is that we can map individual vertex resolution native assessments. So what that means is we put the new subject in the scanner, collect a brain MRI, do the pre-processing. Later on, knowing their covariates, things like age and sex, we can come up with a map essentially detailing where in their brain they might be deviating from what we consider normal for their age and sex. And we thought, what would be a good application to test that this method actually works and it can help us have new neuroscientific findings? And we thought of Alzheimer's disease, given that previous literature reported significant and large-scale cortical atrophy happening in Alzheimer's. So essentially, after training a model on healthy individuals to learn the trajectories of cortical thickness, we utilized the transfer learning capabilities of normative models in general to essentially fit the model into a new data set that was a clinical data set from the MACC harmonization data, and it included 3 cohorts, basically, healthy controls, people with mild cognitive impairment, as well as those with Alzheimer's disease. And we wanted to essentially see if the method can reveal stuff for us. And the findings were twofold. First, we looked at population level findings. We looked at whether the cortical correlates of Alzheimer's disease, and particularly the cortical correlates of cognitive impairment, could be recovered using this method. So we could replicate previous works, essentially showing a widespread cortical atrophy happening in those with Alzheimer's disease in panel A. So all those red regions are significantly thinner than what's in a normal population. But we were also able to associate that localized thinning with cognitive ability, and we noticed that particularly in the temporal pole and in the parahippocampal gyri, cortical atrophy in those regions correlated with cognitive impairment measured by the MMSE cognitive score. And later on, we were also able to examine and tell that it's actually the most extreme thinnings that are the best predictors of atrophy. So the average thinning is not as predictive as the extreme cases. So individuals that have parts of the brain that are really thin, and the number of vertices that are very thin. So more than like 2 standard deviations from the mean. And after that, we were thinking, okay, these are all group level findings. What can we tell about the individuals? And we looked into the individuals and we saw, wow, there's a lot of heterogeneity. For example, 2 people shown here both have the same diagnosis, the same level of cognitive impairment, similar age and sex. However, when you look at their cortical thinning, cortical atrophy patterns, they are somewhat very different. Not similar to the group level map that we saw earlier, and not similar to each other. Interestingly, our approach can also map these at the level of native subject, so either at the cortical surface projection or at the native MNI space. So this can potentially be something that a clinician could have a look at early on to see those markers of extreme atrophy. So we wanted to see if this heterogeneity that we're seeing in this example is actually an exception or kind of the rule. So we hypothesized a few different possible ways in which individuals could be different. The interdividual variability in the normative assessments could have been out of random. They could have been group-based, so people who have Alzheimer's just have similar types of atrophy. It could have been a progression severity-based pattern, or it could have been that those with Alzheimer's are just having more different brains, not just to healthy ones, but also amongst themselves. And that's actually what we found in our empirical data. Those that have Alzheimer's, not only are they different from people who are healthy or with people who are with a mild cognitive impairment, they're also very different amongst each other. And we try to project this into a latent heterogeneity landscape, if you will, to try to make sense of this, to see if this is possibly subtypable. And the way we see our results kind of tells us that subtyping may not be the way clustering will give us cluster, but when we look at this landscape, it's somewhat so scattered. And when you walk around this landscape, different parts of the landscape show different patterns of atrophy. There are actually different cases of Alzheimer's happening in different parts of this landscape, and depending on where you go, the patterns are different. There are some Alzheimer's disease patients that have no atrophy, which was kind of interesting at the time. And more notably, if we zoom back again to the group level, healthy individuals tend to be centered around what's considered a normative center of mass. Those with mild cognitive impairments are further deviated, and those with Alzheimer's disease show that on average, show the most level of deviation, but also towards different directions in the landscape. And essentially, the idea is, as you walk and traverse this landscape, you see different types of cortical atrophy, and our aim is that in future we could link this to possibly different dimensions of symptoms that are correlated with Alzheimer's disease and possibly even with comorbidity maps of Alzheimer's. But yeah, that was as much as I could fit into the slides. So I should, first of all, thank all the collaborators that have helped me in this project. This has certainly been a team effort, and I'm just presenting on behalf of a big group of researchers. And I would like to thank you all for listening and invite you to come visit my poster tomorrow if you'd like to discuss more about this. Thanks so much.
Thank you. It's fascinating work. Thank you. Do we have any questions from the audience?
That was a beautiful talk, so thanks so much for giving it. So I'm just curious what you think is the spectral component kind of contributing to all this. Is it just that it gives you a sort of faster computation time to sort of get at the same thing that you would get at with some sort of like cortical thickness measure, or do you think that there's more to it than that? And I have a follow-up question, but I'm just curious there.
Yeah, so there are two things. Mainly, yes, I think it's just the way to do things more efficiently. So something that would take, let's say, 100 minutes would just take a minute, and that would give us the ability to derive the charts for more measures across larger populations and higher resolutions. But at the same time, when looking at the data, those higher frequencies are usually things that jitter or are really fastly changing along the cortex. So it's an assumption that those things are probably contributed sources of noise. So maybe it's a good idea not to model them in our charts.
Yeah, okay, and I was kind of going towards my kind of follow-up question, which is if you just use any form of dimension reduction, because I think you said you used like 1,000 components or something. If you just use PCA or something like that, do you get a similar speed up? Are the results as sensitive or maybe not as sensitive? Just curious if you've looked at that.
My assumption is as long as the dimensionality reduction has some basis that is valid, I think it should work pretty similar, if not exactly the same. However, we went with the structural connectivity eigenmodes because the literature suggests that particularly the way these propagations happen in the cortex in different pathologies, they're usually mediated by the structural connectome. So if one region is affected, those regions that are structurally connected to it will be affected next. So that was kind of the rationale for using this, but I would imagine that from an information theoretic point of view, changing the basis wouldn't make too big of a difference.
Sure. Thanks so much. Beautiful talk.
Do we have any other questions from the audience? No? I have a quick question. I'm just wondering, particularly in relation to Alzheimer's disease, rather than looking at whole brain, can you kind of hone in on specific areas, for example, medial temporal lobe, and look at the dynamic changes and variability among individuals in regions that we know are affected very early in disease progression?
Yes, well, so the method allows for like zooming into the level of vertices, and we can even actually do it per individual. So an idea that we are like contemplating right now is we could maybe use resting state functional MRI to drive networks. And then if we have a subset that has, let's say, a language deficits within our AD sample, maybe looking at the normative deviations within their language network would make more sense. And yeah, I think that this flexibility that it has might eventually translate into something that makes a personalized treatment or a personalized approach towards treating disorders like dementia.
Hi, beautiful slides and presentation. I wonder whether you have explored the approach in longitudinal data, and so you can track actually progression within subjects.
That's actually what we're doing next. Thanks so much for your question. Yeah, that's kind of the hope. We want to be able to, let's say, look at the normative model of someone who's not yet diagnosed with dementia and be able to predict that they're going to get dementia in 5 years. That's kind of the holy grail for us.
That's very important. Thanks.
Okay, all right. Join me in thanking Sina for a wonderful talk.